Hi, thank you for giving me the opportunity to present with so talented woman. I am Valentina Provoste, a biotechnology student in the current genomic advanced I will present. Sophora toromiro is an endemic tree of Rapa Nui, which is currently extinct in the wild. Despite this, it still persists in botanic gardens, a private collection around the world. It is considered the sacred tree of Rapa Nui, and its reintroduction means the return of its heritage culturate. The main problem for its conservation and reintroduction is the authenticity of the specimens presented, because the existence of hybrid lines leads to misclassification of the species. Based on previously antecedent regarding the chloroplast genome of the S. toromiro and other 4 specimens of Sophora, it is suggested that the TOR2 could correspond to genuine S. toromiro, so that not hybrid. My overall objective is design and validate molecular markers for the development and detection kit that discriminate between Sophora toromiro and its hybrid. The methodology starts with the collection of biological materials of the 5 Sophora species, geolocated and phenotypically identified in the National Botanic Garden and stories and minute. The DNA extraction was made with the Journal Gem Plant ACB mini kit using 100 milligrams of leaflets measured with liquid nitrogen and following the manufacturing instruction. They were sequenced de novo through next generation sequence with the platform Illumina and track a bioinformatic pipeline to complete genomics of all samples were assembled. Subsequently, this data were used to perform comparative genomic and phylogenomic analysis. Search for molecular markers that allow the differentiation of Sophoras. Result: in Tab 1, observe the evaluation of quality of the 5 whole genomics assemblies of Sophora specimens made of de novo using bioinformatic tools, SPADES and QUAST. If you see TOR1 and TOR2 correspond to Sophora toromiro, TOR3 and TOR4 correspond to hybrid lines, and TOR5 correspond to Sophora macrocarpa. And you see the coverage, the number of counting, length, and percentage of GC and their respective EC 50. Figure 1 shows the number of nuclear microsatellites found in the 5 Sophora genomes and the total number of microsatellites obtained after filtering them with a coverage greater than 10. In Figure 2 shows the distribution of the mono, di, tetra, penta, and hexanucleotides in the 5 Sophora genomes. For naked and molecular markers nuclears, I used the unique microsatellite found in each specimen to be used, and showing Venn diagram in Figure 3. For the molecular markers chloroplastidials, I based on chloroplast genome date, show a phylogenomic tree using all the chloroplastic genome of the 5 samples. It is observed that one of the samples previously labeled as Sophora toromiro, so TOR1, is grouped together with the hybrid individuals. On the other hand, TOR2 is separated for the rest of the analyzed individuals, reaffirming its ancestral quality. With this information, we proceed to generate the chloroplast molecular markers in Figure 5. Visualization of the genomes in concentric cycles, showing the hypervariable regions were identified among the Sophora samples represented by white lines on the map, the sector corresponding to gene and EG is selected for all primers. The conclusion of this work is it was possible to identify nuclears and chloroplastic molecular markers or 5 space Sophoras that will allow the development of the toromiro certification systems to distinguish it from its hybrid. We are working in generate a detection kit that allow to reinsert a stable population in Rapa Nui. And I hope that bioinformatics will help you to recover part of the plant heritage of Rapa Nui. Thank you, and thank you so much to CONAF, Botanic Garden National, and Genome Mayor.